The overall goal of this procedure is to semi-automatically segment complicated volumetric datasets of a biological nature.
This method can help answer key questions in the field of structural biology by allowing for the qualitative and quantitative analysis of volumetric data.
The main advantage of this technique is that it greatly reduces the complexity and therefore the time to segment three-dimensional data.
Though this method can provide insight into biological datasets, it can also be applied to other fields such as materials science.
This software uses a different workflow as compared to other segmentation programs, which can cause initial difficulties when first using SuRVoS. Therefore, visual demonstration is critical to understand this new workflow.
Segmentation of large, well-defined regions using model training is demonstrated on a phase contrast X-ray tomography dataset. To begin this procedure, launch the SuRVoS Workbench. Click the Open Dataset button and select the data file to be segmented in the resulting pop-up. Choose an appropriate orientation for the dataset. Next, choose a folder in which to store the workspace and the associated files. Click the Load button. In the Select ROI tab, input the X, Y, and Z start and end coordinates for a region of interest and click Add. Once a section is added, make sure it is selected by checking the box to the right. After this, in the Features Channels tab, use the drop-down menu to choose a feature and add it to the queue.